The next series of discoveries, after we had purified this receptor, was to do what's called cloning the gene. Okay, that allows us, because of the work on DNA, which we've been hearing a bit about at this meeting, and of course by the 80s, this was the era of recombinant DNA was picking up steam, all based on Jim Watson and Crick's original discovery from, I guess, the 60s about DNA. So we were able to ultimately clone the gene for this one particular receptor and thereby deduce its complete amino acid sequence. And when we did that, we made a remarkable discovery. And the discovery was it looked just like another molecule. And that molecule is called rhodopsin. And rhodopsin is the molecule in the eye that allows you to see. And when we saw that, this was in 1986, I realized immediately that, I'll bet there's a huge family of receptors that all look like this. In a sense, rhodopsin is a light receptor. And it looked just like what's called the beta-adrenergic receptor, which is one of these adrenaline receptors that I was studying. And I said, if these two, so disparate in their function, look alike, what about receptors for histamine, serotonin, dopamine, you name it? I'll bet they all look alike. So using the techniques that we had developed, very quickly over the next few years, we were able to get the genes for about 10 or 12 of these different receptors. And they all looked the same. I mean, they have distinct sequences, very close though. I mean, you might have 60, 70% of all the amino acids would be the same, but enough were different that they did different things.